My name is Bessie Kankia, and I'm a scientist at Ohio State University. I'm mainly investigating nucleic acid quadruplexes and trying to develop various biomedical tools and assays based on these quadruplexes. But however, my interest in origin of life goes way back to my student years when I realized that the universal genetic code had to be a product of evolution and could represent useful fossil to understand emergence of life. In 1994, I published this paper which suggested that the genetic code was initiated by interaction of polyguanine with the simplest amino acid, glycine, and then by step-by-step incorporation of other nucleotides and amino acids was transformed into the universal genetic code. However, at that time, I couldn't understand how polyguanine could carry the very first information, which is really a requirement to initiate Darwinian evolution. So this paper was not supported really by some solid arguments and actually represented a speculation. In 1997, I came to the United States, got involved in more practical biomedical research, and basically forgot about my these code evolution ideas. However, during COVID pandemics, when I got lots of time to think about everything, including this paper, I realized that guanine-rich sequences, including the homopolymer poly-G, not only can be used as the driving force or building blocks of nanostructures, but they are programmable without any involvement from the complementarity principle. So polyguanine is not just an artificial and boring homopolymer, but it is able to carry a specific information. Based on this discovery, I'm developing quadruplex world hypothesis, which with surprising simplicity addresses all origin of life questions. Main aspects of this quadruplex world hypothesis are published in these three papers, and today I'll be talking mainly about these papers, but with deeper analysis which took place during the last year. I would like to start with a quote from Albert Einstein, which gives us a good advice how to solve big scientific problems. He says, "We cannot solve our problems with the same thinking we used when we created them." To better understand this quote would be useful to discuss one of the biggest questions on Earth, why we have day and night. This 19th century etching marks the middle centuries when people thought that Earth was flat, and to find an answer one needed to walk long enough to reach the horizon or the junction of the Earth and heaven. If you could reach horizon, solving the problems would be very easy. Just look behind the heaven like this guy is doing and find all answers. Of course, within the flat earth thinking, the day and night questions would not leave field of curiosity or mythology. But during Roman times, Roman philosopher Ptolemy transformed this curiosity into a scientific problem by suggesting that the Earth, Sun, Moon, and all other planets were spherical objects and are revolving around the Earth, which was considered, Earth was considered as a static center of the universe. Of course, we know that this is just an impression due to the fact that Earth is spinning around the axis. However, besides this incorrect assumption, Ptolemy and others would suspect that there was something wrong with this model. The geocentric model couldn't explain the phases of moon or retrograde of planets. All these problems were solved later by Copernicus during Renaissance, when he suggested heliocentric model. This is a final model tested by modern telescopes. So looks like what really Einstein tells us. It tells us that initially the question represents a simple curiosity leading to nowhere. Then some kind of thinking transforms the curiosity into the scientific problem. But this thinking is incapable to solve the problem. To solve the problem, we need to change the thinking or approach. So Einstein does not say that we need to step by step increase our knowledge, and then from this bigger knowledge we can solve the problem after some time. In this case, it took us 2,000 years. No, what Einstein suggests, we can solve the problem very next day, but we need to change thinking or scientific approach. Now let's return to our main question, origin of life. Before discovery of the complementarity principle by Watson and Crick almost 70 years ago, The origin of life question belonged to a field of curiosity or mythology. Without knowing how cells divide and what was the carrier of the information, we could not make any scientific scenarios. As soon as the complementarity principle was discovered in 1953, and we realized how cells divide and transmit the information, very soon after that, in 1960s, the RNA world hypothesis was suggested by Crick himself and others. The RNA world hypothesis suggests that RNA can represent a primitive life form by reproducing itself and establishing the genotype-phenotype relationship without any external help, including from the proteins. Both functions are achieved through the complementarity principle, which allows RNA to store information and program functional 3D architectures, such as tRNAs and ribozymes. However, the RNA world has many issues and paradoxes, and so far all these problems have been addressed within the complementarity principle, and however, without any really significant success. In other words, to solve the origin of life problem, people still use the same thinking which was used during creating the problem. Here I suggest changing the approach by initiating the process with the quadruplexes. So main goal of my talk to convince you that this approach solves all RNA world problems like the heliocentric thinking solved problems of the geocentric thinking. First of all, I would like to emphasize that origin of life problem is not as mysterious as origin of universe, where we have no idea what was before the Big Bang. Origin of life actually represents a missing link between these two well-characterized systems, the primordial soup and LUCA, or the last universal common ancestor. The primordial soup is a well-understood system. All components of the biopolymers, such as amino acids, nucleic bases, sugars, and phosphates, can be produced during abiogenesis. And LUCA is the ancestor of all living creatures on the Earth and represents a simple but autonomous cell based on the same universal genetic code. So any valid origin of life hypothesis should suggest a fully tractable, experimentally testable, and logically understandable scenario by rules of standard physics and chemistry. The missing bridge consists of at least two segments. First is a pre-Darwinian or chemical evolution, and the second segment is an early stage of Darwinian evolution. So, question of origin of life is almost identical to origin of Darwinian evolution. So, Darwinian evolution does not exist yet and must be bootstrapped within a chemical evolution by some polynucleotide. Idea of bootstrapping comes from the Western boots and suggests that the very first system has to use the straps to lift itself up or elevate itself from the chemical evolution into the Darwinian evolution without any external help. It's really interesting that Darwin talks about the bootstrapping process. Of course, he perfectly understands that the evolution cannot initiate process. It is a passive observer. It doesn't produce anything. It just selects the most advantageous life form. So in these quotes, Darwin emphasizes that the very first step, which initiates the endless life forms, must be a very simple process, but it appeared not by the evolution, but by some wholly unknown process. So evolution, a wheel, for instance, starts here, and the very first version of the wheel must be created by the bootstrapping operation. So a valid origin of life hypothesis must obey the following rules and principles. First, bootstrapping had to be a chain of scientifically explainable causal events, or just standard physics and chemistry should be able to explain all this process. Second one, the bootstrapping should not involve Darwinian evolution. As I mentioned in the previous slide, it emerges after the bootstrapping, so it really didn't exist yet. Third, product of the bootstrapping should be functional, at least at a minimum level. So if we consider, let's say, previous examples, that the evolution of the wheel, the very first wheel had to be able to rotate around its axis and roll, and by this make movement of heavier loads easier. Or in our case, we believe that the very first system in biology was ribosome. The very first proto-ribosome had to polymerize amino acids on a nucleic acid scaffold. And the last principle is a Darwinian continuity principle, which means that Darwinian evolution after the bootstrap should comprise of incremental Darwinian steps without foresight and miracles. So, what bootstraps Darwinian evolution? Or what happens here at this junction? Of course, Darwinian evolution could not kick in without these two processes. We need the replicase to create heredity and divide the cells, and we also need the ribosome to establish the genotype-phenotype relationship, or GPR. The question is, which came first? Initially, let's characterize both of them and discuss which one had more chances to emerge during the chemical evolution. The replicases are catalyzed by the protein enzymes. However, emergence of the protein enzymes before ribosomes is doubtful. Any accidental protein emerged outside of the translation apparatus had no chance to become part of the heredity. So, proteinaceous nature of the replicases is hard to explain by the replicase first approach. However, other properties could easily emerge during chemical evolution. For instance, direct Watson-Crick information enforcing mechanism, which is really based on direct physical binding between nucleic bases. In addition, all extant replicases have reducible complexity, not only truncating different domains, but even complete removal of the enzyme does not stop the reaction completely, since it is sterically possible. And as all other protein enzymes, the replicases are true catalysts, just increasing the rate of the already running reaction. So they are catalyzing reaction which already has some background activity. Now let's talk about the ribosome first approach. The ribosome is catalyzed by the ribozyme, which supports its abiogenic origin, but all other properties are too complex. For instance, ribosome uses a genetic code as an information encoding decoding system. Second, they require adapters or tRNA. And third, ribosome is irreducibly complex. Even deletion of outside proteins, which are shown here in blue, completely stops the reaction. This is because of placing of two tRNAs, as shown in this figure, on two adjacent triplet codons is sterically impossible. It requires bending of the messenger RNA and enforcing the reaction by the ribosomal RNA. So ribosome is not just a typical enzyme which accelerates reaction, which already has a background activity. It also is not an organelle, so it's really unique, which should be due to its de novo emergence. If we assume the replicase first approach, which actually leads to the RNA vault hypothesis, we need to assume that initially replicases were catalyzed by the ribozymes. However, here are two major problems. First, at some point during the evolution, the ribozyme catalyzed or ribozyme replicases need to transform into the protein replicases, which seems highly unlikely. Such a dramatic transformation is uncharacteristic for Darwinian evolution. Second, if the origin of life initiates by the replicase, the emergence of the ribosome had to be a Darwinian process. And since ribosome does not have any immediate selection value, emergence of the ribosome becomes clear foresight. Also, explanation of such complex machinery by an acceptation would be also very hard. Now, let's consider ribosome first approach. If we consider this approach, we need to find a stereochemically possible and very simple proto-ribosome, which has background activity. At first glance, this seems impossible due to the extreme complexity of the ribosome, but quadruplex world hypothesis really offers a perfect solution. First, let's quickly discuss replicase first or RNA world hypothesis. This is a timeline of events taken from the Joyce paper, which is very general. Just tells us that RNA world sits somewhere between pre-RNA and extant biology. So we can consider the RNA world at a given period of time. Even we can conduct some supporting experiments using today's modern molecular biology techniques. However, it is absolutely unclear how it arrived from the prebiotic chemistry or how it will transform into the extant biology. So it is virtually an isolated system. This is a more detailed scenario of the RNA world from a recent paper which reveals its paradoxes and problems, and they are shown, paradoxes and problems are shown in red. Production of the very first information, or the ribozyme replicase, is a random process. All kinds of molecules had to be synthesized, or the random pool of molecules should be created first, from which the replicase had to be selected later. Formation of the pool is merged with the following problems: breaking the chirality or selecting only nucleotides with D sugars. Another problem is cyclization, which terminates RNA polymerization. RNA is pretty flexible and polymerization requires scaffolding or some mineral crystals. Another problem is constant hydrolysis, since RNA easily degrades in aqueous solutions. Also, the random nature of the polymerization requires astronomical pools or molecular libraries, which are one of the biggest paradoxes, and I'll talk about this one in a minute. Then, of course, we have misfolding problem. At this point, also RNA world assumes existence of all kinds of ribozymes, which brings to another violation. Natural selection is required before emergence of the Darwinian evolution itself. Then, the selected structure must be amplified, which is accompanied by two paradoxes: functional duality and the Huygens paradox. The functional duality paradox requires that the same 3D architecture must be unstable to be invaded and replicated, and at the same time it has to be stable to perform self-replication. And the Huygens paradox is due to the exponential nature of the template-guided replication, which accumulates mutations exponentially and leads to the genetic meltdown. To avoid this meltdown, the replication requires not only sophisticated extant protein polymerases, but even error-correcting enzymes. And finally, transformation of the ribosome-governed cells into the ribosome-based cells, or functional takeover from ribozymes to enzymes. Now let's consider the ribosome first, or quadruplex world hypothesis. The recognition element of the quadruplexes is G-tetrad, which is stabilized by 8 Hookesten hydrogen bonds, cation chelation, and strong stacking interaction between the G-tetrads. The structure requires specific cations. For instance, the most favorable cation is potassium, and to avoid the quadruplex formation, one needs to use lithium or cesium. DNA quadruplexes are polymorphic due to their ability to adopt both syn and anti-glycosidic bond conformation. Syn conformation are shown in pink and anti in blue. The syn conformation is required for the anti-parallel structures. As you can see from this figure, most of the DNA structures reveal the anti-parallel topologies. However, RNA can adopt only anti-conformation suitable for only all parallel topologies, which is shown here. So RNA quadruplexes are monomorphic with only the all parallel topology, using the Z-shaped or double chain reversal loops. Now let's discuss this all parallel topology in more details. Here is the structure formed by the homo-oligomer G15. Left panel shows the right-handed helix with 30-degree helical turn, and the right panel shows its unfolded two-dimensional map. Monomolecular RNA quadruplex can be folded only in this architecture with only 3 G-tetrads and Z-shaped loops. And we call this structure quadruplex domain, or QD. The loops can be formed by any nucleotides, including guanines. So G4, G8, and G12, shown in black, are used in the loops. It is important to emphasize that monomolecular RNA quadruplex is incapable to employ any other folding pattern, and as a result, long polyguanine has a domain assembly. It forms many QDs in a row. This is the origin of life scenario suggested by the quadruplex world. It is fully tractable and experimentally testable. Initially, free guanine monomers self-organize in the G-tetrad stacks and easily ligate. The structure is very stable and can be used as a purification tool upon temperature cycles. Short oligomers arrange into the G wires and further polymerize, and when they reach lengths of 15, the quadruplex domains start folding and are ready to initiate the proto-translation process. Now let's discuss each step separately. This slide shows self-organization of the free guanine monomers and the initial ligation. The G-tetrads form right-handed crystalline structure, which was demonstrated in these papers. The helical properties of the crystalline structure are the same as the quadruplexes formed by polyguanine strands. As a result, the reaction moieties of the free guanines are perfectly juxtaposed for the ligation. So ligation was demonstrated in these papers. So the very first step of the quadruplex world scenario addresses several major problems of the RNA world. In the right-handed crystalline structures, adjacent guanines can be ligated with only D sugar. So problem of chirality is solved by the preference of the free G-tetrads fold into the right-handed structures. Also, we do not have the cyclization since the tetrahelical structure is very rigid and stable. So quadruplex world does not require outside scaffolding from any clay minerals. It is a self-scaffolding system. Also, due to the high stability of the structures, ligation overcomes the hydrolysis. The problem of the astronomical pool and other problems and paradoxes I'll discuss later. Another advantage of the quadruplex world is that the temperature cycles can be used for purification. Free guanines, these black dots, need to self-organize in the crystalline structures in the presence of various prebiotic compounds. So these structures most likely will be contaminated by other nucleotides or compounds. Contaminated parts are shown as pink discs. Adjacent pure G-tetrads would ligate correctly, shown as blue arrows, while the contaminated parts would terminate the polymerization or form incorrect oligomers, shown as the red segments. However, during the heating step, the complex will dissociate, and upon cooling step, only correct G oligomers would re-enter the G wires due to their higher stability. In other words, purified G wires will be already formed at relatively high temperatures before impurities could re-enter the structure. This is possible because oligoguanines can slip against each other in the G wires and form and reform same G wires with completely different strand combinations. This is, of course, impractical for the RNA duplex. So this slide really demonstrates three important things. First, quadruplex world can have the purification tool. Second, the problem of compartmentalization also can be solved because after several temperature cycles, whole reservoir or primordial pond would transform into a large communal cell with polyguanine surrounded by other prebiotic compounds. And third, the production of polyguanine is a template-free and very robust process. Now let's talk about the G wires and the ligation. G wires are well documented in many studies. Here I only show an atomic force microscopy image from this pioneering paper. And ligation of G wires was demonstrated by us, which revealed very efficient polymerization of G10 into polyguanine with the polymerization degree of more than thousands. In this and the three following slides, I'll show different experimental proofs that polyguanine can fold only into the quadruplexes with 3 G-tetrads and single nucleotide loops, or in other words, it employs only 3x1 folding pattern. This NMR study demonstrates that both G15 and the similar construct with T loops are folded in the same structure with already mentioned 3x1 folding pattern. Both molecules are characterized with 12 identical immunoproton resonances. This study demonstrates that while DNA sequence with G4C2 repeats forms anti-parallel quadruplex with 4 G tetrads, each RNA analog forms all parallel topology with only 3 G tetrads and long GCC loops. So, while the molecule has sufficiently long loops, the quadruplex is unable to accommodate 4 G tetrads, despite the fact that stacking interaction is thermodynamically favorable. Another proof comes from the statistical morphology analysis of atomic force microscopy imaging of long polyguanine, which can only be explained by domain assembly of the quadruplexes with 3x1 folding pattern, as shown here. We also performed systematic thermodynamic study of both RNA and DNA sequences. By incorporating the T or U loops at different positions, one can create the quadruplex domains with the different folding patterns. For instance, this sequence, very first one, can adopt only 2x1 folding pattern, or the last sequence has a freedom to employ 4x2 or 3x3 patterns. In case of 4x2 pattern, stability has to be significantly higher than that of stability of 3x1, since the extra G-tetrad stacking should stabilize the structure by 15-20 degrees Celsius. Similarly, circular dichroic signal for 4x2 folding pattern has to be at least 30% stronger than for the 3x1 folding pattern. The study revealed that the sequences with the 2x1 and 2x2 folding patterns do not fold any monomolecular structures. The 3x1 folding pattern has the highest thermal stability and CD signal, so all other RNA sequences with the potential to accommodate 4 G tetrads represent the destabilized versions of the 3x1 pattern. So, the study fully agrees with the literature data and suggests that polyguanine can accommodate only 3x1 folding pattern. Now it's a good time to define the biological information. Biological information can be defined as a base sequence of RNA or genotype that unambiguously programs formation of a functional 3D architecture or phenotype using predetermined set of rules. The main tool of biology which processes the biological information is the translation apparatus or the ribosome. The information encoded into the messenger RNA dictates production of the functional protein using highly sophisticated set of rules, the genetic code. So the ribosome represents the main tool to establish the genotype-phenotype relationship. Another tool is the folding of RNA into the functional three-dimensional architecture, such as tRNA or ribozymes. It uses the complementarity rules based on the direct physical binding between the bases or base pairing, anti-parallel strand orientation, and U-shaped loops. This tool is partially used in the extant biology and represents the main information encoding mechanism in the RNA world. And here is the third information processing tool, so far unknown, quadruplex folding, which employs G-tetrads, parallel strand orientation, and the Z-shaped loops. So G15 encodes folding of one quadruplex domain, or if you have G30, like it is shown here, it is programmed to fold into two quadruplex domains. So polyguanine is not just a dull homopolymer incapable to carry any information, but it is a single information carrying material which ideally fits to the origin of life. Of course, here emerges the question, which might help us to understand the most mysterious aspect of life, triplet nature of the genetic code. Is there any link between triplet nature of the quadruplex domains and the genetic code? This is a section from the Crick's seminal paper with the self-explanatory title, Why a Triplet? Here he absolutely correctly suggests that the code must have been basically a triplet code from a very early stage. Of course, any modification of the basic rules of the genetic code would have a little effect. Then he assumes that the reason of the trinity must be due to the invariant structural properties of the adapter, serving as a measuring stick for the triplets. And at the end of the section, he hypothesized that measuring stick may be the diameter of RNA duplex, since the tRNA represents the adapter in the extant biology. However, tRNA or any other adapter based on the double helix would not meet the requirement, since the RNA duplex diameter, 2.3 nanometers, is significantly larger than the length of the triplet, which is around 1 nanometer. In addition, folded duplexes have negatively charged surfaces, so placing two tRNAs on two adjacent triplexes is physically impossible, while the quadruplex domains perfectly fit to the job. The QD represents an ideal measuring stick and determines the triplet nature of the genetic code. Quadruplex domain has multifaceted surface and could play a role of proto-tRNA and single-handedly conduct the translation. It has exposed bases to bind to messenger RNA. The end G tetrads are open for stacking interactions, and the loops can form the pockets for the amino acid binding. So quadruplex domain could ideally play a role of the proto-tRNA, and with the incremental steps transform into the extant tRNA. More details about this one can be found in this paper. So this is a hypothetical mechanism of the primitive ribosome. The charged QDs will bind to the adjacent triplets and facilitate peptide bond formation between the amino acids. So the quadruplex domain establishes rules of the genetic code: triplet codon lengths, non-overlapping codons, and commaless reading. So the quadruplex word logically explains one of the most mysterious emergence questions, emergence of the ribosome and the genetic code. This gloomy title I took from the Wolf-Kunin paper. Due to the scary specter of irreducible complexity, some scientists even consider origin of ribosome beyond the scope of scientific research. As I mentioned earlier, even deletion of the exterior proteins, which are not involved in the catalysis, completely inhibits the reaction. And this is due to the stereochemical impossibility to place two tRNAs on two adjacent codons. So the ribosome can be compared with this Roman arch. Removal, even a single stone, results in its total collapse. However, we know that the arch was built on the scaffold, which was removed later. So scaffold is gone, but we can envision the scaffold because the inner perimeter of the arch is determined by the scaffold. So the architectural scaffold does not disappear without a trace, and looks like this is true for the ribosome too. The quadruplex-based proto-ribosome can represent the scaffold on which the extant ribosome was built. And after introducing the complementarity principles, the scaffold is gone. However, the perimeter or messenger RNA is conserved, in which information encoding rules must be the same for both the proto and the extant ribosomes. Here I demonstrate how quadruplex world addresses all problems of RNA world. These four problems I covered earlier. An astronomical pool will be discussed in the next slide. Misfolding is characteristic for only double helical RNA and completely missing in quadruplex world because poly G has strictly defined folding patterns, so it does not have any other folding patterns. And also RNA world requires natural selection before creating the very first information. So we got this overlap, which again quadruplex world is free from this problem. Functional duality in the quadruplex world does not exist, since genotypes are produced in G wires, and as soon as the degree of polymerization reaches 15, quadruplex domain is folded, which is very stable and never re-enters G wires back. So In quadruplex world, production of the genotype and the phenotype are two separate processes. Eigen's paradox in quadruplex world does not exist, since quadruplex domain production is not the template-guided process and the mistakes don't accumulate. If a construct incorporates other than guanine, it's not able to form QD or re-enter the G wires, and as a result, it's removed from the genotype production process. Also, since the ab initio system is a protoribosome, proteins can involve in the evolution at very early days to start working on the replication. So there is also no functional takeover of quadruplex world, since the protoribosome immediately starts working on protein production. So nucleic acid protein world emerges at very early stage. Now let's discuss the astronomical pools. One of the biggest paradoxes of the RNA world is that it requires gigantic sequence spaces or the libraries. For instance, an 80 nucleotide long RNA library would contain 10 to the power of 48 of molecules. The weight of such library containing only a single copy of each sequence equals to the mass of Earth. The weight of 130 nucleotide long RNA equals to the mass of whole universe, and the weight of the 1800 nucleotide RNA, shortest RNA required to initiate translation replication tandem, is 10 to the power of 1004 times larger than the universe. The libraries are astronomical because RNA world employs the complementarity principle as an information encoding mechanism. The complementarity is a multi-component or multi-letter system, and this is what creates the astronomical libraries. We could try to reduce the library by removing AU base pair, but this really does not help. Numbers are still astronomical. However, further reduction to the single letter system drops the astronomical number of messages to the single message. This seems as a logical fallacy, since a single letter system eliminates the complementarity. However, poly-G is a programmable molecule without complementarity and has strictly defined and length-independent folding pattern, ideally fitting to the role of the very first information which self-emerged during the chemical evolution. So, any system requiring more than one component initiates the process with the astronomical number of the messages and would require the natural selection before arrival of the Darwinian evolution. So, only way to initiate process with a single information is a single letter system or a homopolymer. So, polyguanine represents a polymer with the informational singularity or zero entropy, preceding Big Bang of the complementarity principle. Why we don't see folded quadruplexes in vivo? After introducing the complementarity principle and replicases, the quadruplexes became easily invadable targets by the complementary strands. Therefore, despite numerous G-rich sequences in all genomes, we don't have clear evidences of the folded quadruplexes in vivo. This echoes with Darwin's assumptions that initial molecules would be instantly devoured or absorbed by later molecules, in our case invaded by the complements and transformed into the duplexes. However, we have several indirect clues on the existence of the quadruplex world. For instance, numerous quadruplex folding segments in all genomes and long telomeric repeats. Also, the quadruplex scaffolding of the extant ribosome, which initiates the genetic code as collaboration of GGG codon and glycine and establishes the grammar rules of the code. Another very interesting clue comes from the aptamers, which often employ G-tetrads as a structural motif. The aptamers are functional three-dimensional molecules produced and selected from the man-made nucleic acid pools. So, emergence of the functional molecules during abiogenesis and the aptamers in the man-made pools conceptually are similar. Interestingly, one of the first aptamers shown in this paper was based on our quadruplex domain. This further emphasizes how easy it could be to produce the quadruplex domains abiogenetically. So quadruplex domains seem to be too straightforward and important to be ignored by abiogenesis. At the end, I would like to show again the bootstrapping process where three guanines form the G-tetrad, uptake cation in the middle of the structure. It is important to know that guanines are not able to form any other complex. After G-tetrad formation, G-tetrads starting stacking to each other with a 30-degree helical turn, ligate easily, and after reaching needed polymerization degree, which is 15, fold into the quadruplex domain, and finally initiate the protoribosome. So the bootstrapping process is causally deterministic process where each state represents the effect of its past state and causes the following state. So as soon as guanine emerges, the blueprint of the bootstrapping process leading to the protoribosome already exists. So thanks for listening, and I'll be happy to answer any comment posted under the video or sent to this email.